I'm
你这
Jiadong
个
Wang
OK。
from Peking University. First, I would like to thank the organizers of this meeting. Today, I'm going to talk about a post-transcriptional process modulates cyclical expression of BRCA1. BRCA1 is one of the most important tumor suppressors, which is inactivated by mutation in familial breast cancers. BRCA1 gene mutations covers about 60 to 80% lifetime risk of the development of breast cancers and ovarian cancers. However, most sporadic basal-like breast cancer without BRCA1 germline mutation but decrease BRCA1 expression. Dysregulation of BRCA1 expression contributes to about 40% sporadic breast cancer and more common in ovarian cancers. Now we know the anti-tumorigenic functions of BRCA1 are mainly through maintaining genomic stability. BRCA1 plays essential functions in cell cycle regulation, homologous recombination repair, the double-strand break repair pathway choice, and replication fork protection. In G1 phase, the double-strand break is mainly repaired by phase 3 BP1-mediated non-homolog and draining repair pathway. In SG2 phase, the double-strand break is repaired by homolog recombination pathway. BRCA1 could antagonize phase 3 BP1 RIF1 complex to promote homolog recombination in SG2 phase. When dysregulating BRCA1, the double-strand break also could be repaired by non-homolog and draining in SG2 phase, which could promote chromosome translocation, genomic instability, and tumorigenesis. In other words, lower BRCA1 expression in G1 phase is necessary to avoid excessive DNA recombination-induced DNA damage, and higher BRCA1 expression in S phase is also important for accurate DNA repair. Therefore, sudden increase of BRCA1 expression from G1 phase to S phase is necessary to prevent genomic instability and tumorigenesis. In fact, the BRCA1 expression are reported to change along the cell cycle. The BRCA1 protein level are higher in SG2 phase and lower in G1 phase, and the BRCA1 mRNA level also increased from G1 to S phase. So we're wondering which factor determine the cyclical expression of BRCA1. Understanding how regulation of BRCA1 and the physiological condition is the foundation to understand the pathological dysregulation of BRCA1-induced tumorigenesis. We studied this question from transcriptional level, post-transcription level, and post-translational level. Firstly, consistent with the previous study, we also found the MR level of BRCA1 gradually increased from G1 to S phase. However, the BRCA1 pre-MR level, which reflects the transcription activity of BRCA1 gene, is decreased from G1 to S phase. Therefore, we further detected the BRCA1 transcription activity by POL2 chip assay. We found the association of POL2 with BRCA1 promoter is also decreased in S phase. So transcriptional regulation could not explain higher BRCA1 expression in SG2 phase. Next, we detected the BRCA1 protein stability in G1 or S phase. Surprisingly, both the endogenous BRCA1 protein or ectopic expressed BRCA1 protein show a longer protein half-life in G1 phase compared with in S phase. That means the post-translational regulation either could not explain the higher BRCA1 expression in S phase. Last, we checked the BRCA1 mRNA stability between G1 and S phase. Interestingly, in different cell lines, the BRCA1 mRNA half-life is significantly longer in S phase compared to in G1 phase. So the BRCA1 expression increased from G1 to S phase may regulate it at post-transcriptional level rather than transcriptional or post-translational level. So what happened on BARC1 mRNA between G1 and the S phase? We performed RNA sequencing to analyze the BARC1 mRNA changement. Surprisingly, the alternative splicing of BARC1 changed greatly in different cell cycles. There are more BARC1 D11Q and BARC1 5Q S forms from exon 11 or exon 5 alternative splicing in G1 phase. BARC1 5Q S form have premature termination codon and could be dismissed by nonsense mediated mRNA pathway. The AMD pathway prevents translation of mutant mRNA contain premature termination codons by targeting them for degradation. When mRNA contains PTC, it could recruit UPF1 to activate the AMD pathway and degrade this mRNA. Our previous data shown in G1 phase, the BARC1 mRNA is unstable, and at the same time, there is high-level BARC1 5Q-S form, which contain PTC. To review whether AMD pathway determine the cyclical expression of BARC1, we knock down UPF1 to inhibit the AMD pathway. We found AMD inhibition totally abolished the expression difference of BARC1 mRNA between G1 and S phase. Next, we perform RNA pull-down coupled mass spec to screen the protein regulating BARC1 mRNA splicing along the cell cycle. Biotin-labeled RNA of BARC1 exon 5 and exon 11 were used to pull down proteins from cell lysis in vitro. Remarkably, RBM10 binds to BARC1 RNA with the highest affinity among the splicing factors. RBM10 is a nuclear RNA binding protein and has been reported involved in regulating alternative splicing. We found the BARC1 exon 11 RNA could specifically pull down RBM10 protein, and the RBM10 protein also could specifically associate with BARC1 mRNA, and this association is S-phase dependent. Then we further confirmed whether RBM10 could affect BARC1 cyclical expression. We found that GetRNA-mediated RBM10 depletion significantly decreased BARC1 both in protein level and mRNA level, and this phenotype could be rescued by reconstitution of RBM10 in RBM10 depleted cells. More importantly, the depletion of RBM10 abolished the greatly increase of full-length BARC1 transcripts or protein from G1 phase to S phase. This data suggested that RBM10 is required for the cyclical expression of BARC1. To further study how RBM10 promotes cyclical expression of BARC1, we performed RNA-seq analysis after knocking down RBM10. As a result, loss of RBM10 significantly increased BARC1 D11Q S form and failed to produce full-length BARC1 transcripts. By mini-gene splicing reporter system, we found depletion of RBM10 dramatically increased BARC1 D11Q S form and 5Q S form, and this phenotype could be rescued by RBM10 reconstitution in RBM10 depleted cells. Importantly, after inhibition of the AMD pathway by CHX treatment or UPF1 depletion, the BARC1 mature mRNA level in RBM10 depleted cells also could be rescued. This data demonstrates that RBM10 mediated mRNA splicing promote full-length BARC1 generation and BARC1 expression by avoiding ASMD. Next, we try to identify the upstream signal with controlled RBM10 splicing activity. CTK-mediated phosphorylation plays essential roles in cell cycle-specific events. We found that RBM10 is a CTK substrate, and its phosphorylation level peak on S-phase entry which is dramatically repressed by CDK2 inhibitor. Importantly, CDK2 inhibitor decreased BRCA1 expression depending on RBM10 status, suggesting that CDK2 regulating BRCA1 through RBM10 mediated splicing activity. Mechanically, CDK2 inhibitor suppress BRCA1 full-length transcript generation by inhibiting RBM10 bound to BRCA1 mRNA. Interestingly, RBM10 regulated gene pattern highly correlates with CDK2 regulated gene pattern, suggesting that CDK2 may generally regulating gene expression by phosphorylating RBM10. We further performed a mass spec analysis and identified 3 cellular residues among RBM10 of potential CDK phosphorylation sites. Moreover, only the phosphorylation level of S89 mutant was dramatically decreased by detecting phospho-CDK substrate antibody. We further generated RBM10 S89 specific phospho-antibody, and as expected, the S89 phosphorylation is S-phase specific, and this phosphorylation could be significantly suppressed by CDK2 inhibitor. Moreover, RBM10 phosphorylation mutant could not bind in BRCA1 mRNA, either could not restore BRCA1 expression level in RBM10 depleted cells. Furthermore, the hyperphosphorylated mutant of RBM10 could promote BRCA1 expression even in G1 phase. This data suggested CDK2 mediated RBM10 S89 phosphorylation is important for RBM10 regulate cyclical expression of BRCA1. Notable, we found that G1 and S-phase differentially expressed genes show significant correlation with NMD-regulated genes. To our surprise, about 30% S-phase upregulated genes were controlled by NMD pathway. Importantly, these NMD-regulated genes show more difference between G1 and S-phase and a more rapid change rate. Interestingly, NMD pathway modulated genes were highly enriched in the replication pathway, and the inhibition of NMD pathway affects the replication fork protection and the replication stress, and also decreased replication fork symmetry and normal condition. This data suggested ASMD pathway promotes replication fork progression by regulating DNA replication-related gene expression in S-phase. We also found that depletion of RBM10 decreased 14 HR-related gene expression. Therefore, DNA repair capability was impaired by depleting RBM10, and this phenotype could be rescued by RBM10 reconstitution. Importantly, RBM10 depleted cells displayed hypersensitivity to IR or PARP inhibitor treatment. We further confirm both HR efficiency and replication fork stability were significantly repressed by RBM10 depletion. This data show that RBM10 play important function in maintaining genomic instability. We found there are extremely significant positive correlations between the expression of RBM10 and BRCA1 in either normal tissue or tumor samples. Next, we found that CDK2 inhibitor and PARP inhibitor has synergistic effect to cure cancer both in vitro and in vivo. Okay, let me summarize our findings. Firstly, we found CDK2 mediated RBM10 phosphorylation modulates BRCA1 cyclical expression through ASMD pathway. Secondly, ASMD pathway broadly regulates the expression of cyclical genes such as DNA replication-related genes. Lastly, we also uncovered several potential targets for cancer therapy. I would like to thank my lab members and collaborators. This study is mainly contributed by Nie Chen, 周晓 and 周家栋. Thanks again for your attention.
So, thanks. Any questions?
Hi, Jiadong. Can you hear me?
Yeah, I can hear.
Oh, this is Lin Deng from Shenzhen Bay Laboratory. So I wonder if your model is very. I wonder if it's if it's possible the different isoform use different from UTR for the stability, like the API pathway. I know you this ASNMD pathway. Is it possible a secondary mechanism to regulate the mRNA stability?
Yeah, this is a great question, Deng. Actually, we use the standardization sequencing to verify this phenotype, and we found that the D11Q and the D5Q isoform is very rare to happen at the same time. And we believe the ASMD pathway is consistent with the transcription regulation level. It's a two regulation level to maintain transcription change efficiency between different cell cycle. I think using the three UTR check the mRNA efficiency or stability is required to further answer your question. Thank you.
It's a technical. Oh, sorry.
Excuse me?
It's a cyclical expression type. Disappear if you have the. Wait, I have to change microphone. Sorry. RBM10 SHR to alanine. So this mutant. Do you actually?
Sorry, I didn't hear the question. Did you ask the S98 mutant is required for the RBM10 function, right?
Yes.
Yeah. Actually, we tried the S98 mutant, different mutant. One is abolished the sharing phosphorylation site. The other one is mimic the S-styrene phosphorylation status. Actually, these two mutants has different phenotype. The mimicking phosphorylation mutant could rescue the RBM10 function very well. Actually, use this mutant even in the G1 phase cells, it could increase BRCA1 expression. So we believe that the S89 phosphorylation of RBM10 is required for the RBM10 actually to recognize BRCA1 pre-mRNA and could efficiently to splicing this isoform.
You found RBM10 bound mRNA, do you observe BRCA1 or other mRNA as well?
Oh, yeah, this is a great question. Actually, we using the RBM10 to perform the RIP assay and the sequencing expect the BRCA1 RNA, and we found a lot of other like DNA replication related RNA gene RNA could be regulated or recognized by RBM10 protein.
So any questions from online? Okay. Questions online? So which question? Kind of.
One question from Tengxun, and they ask if BRCA1 mRNA become less stable in other time period other than the S phase. If so, did you test how it could occur?
Yeah, this is a great question. Actually, the BRCA1 RNA in the stability in the S phase, we compared the stability from G1 phase and the S phase. Actually, we also compared in the G2 and the M phase. Actually, in the G1 phase, the BRCA1 is as the same as the G2 and the M phase, but in the G1 phase, is very unstable. And we believe this mRNA instability in G1 phase is due to the PTC. This could be degraded by ASMD pathway. But in G1 phase and G2 phase, the PTC, the mRNA transcription, is very rare in these two kind of phase, and this mRNA relative in stay stable in the G2 and S and M phase, but unstable in the G1 phase. Thank you.
So, because of the time, we move to the next speaker. The next speaker is.
Thank you.
Okay, thanks again. Okay. So the next speaker is Dr. Fei Nongmeng. He is the principal investigator in Shanghai University of Biochemistry and Cell Biology. And welcome.
Hello, everyone. This is Fei Long, and first of all, I want to thank the organizers to give me this chance to present our work. Today, I'm going to present you a story about how mesoscale DNA features can affect the deaminase initiated hypermutation in antibody gene coding sequence. My lab is particularly interested in how the lymphocyte can diversify their antigen receptors to recognize different pathogens in our environment, including SARS-CoV-2. And this process is initiated by programmed DNA damages. At the first step, RAG, it is endonuclease. It can initiate VDG recombination, and at the secondary step, AID, which is cytidine deaminase, it can introduce hypermutations at this variable exon for antibody, or it can produce class switch recombination in the constant region to change IgM to IgE or other isotypes. So with this two-step of diversification, our body can have a full protected antibody repertoire to have a robust immunity. AID is a single-strand DNA cytidine deaminase. It was discovered like more than 20 years ago, and people know it working on DNA, and it prefers to deamine C in the WRC motif. And the best motif actually is AGCT because it's a palindromic sequence have two Cs for AID deamination. But the protein structure analysis show on the surface of this protein there are two paths of positively charged channels. One is for the substrate channel, and another they call it associated path. However, the function is still unknown. In hypermutation, AID activity is specifically targeted to this variable region we call IgV exon. This small exon actually coding Ig domain for antibody. So the CDR region directly binds the antigens. And the framework actually is the green one here is mainly maintaining this protein stability. So basically, if we want to design this system, we want more mutations at CDR, so we have a higher chance to select for a high affinity antibody. However, we prefer to have less mutations at this framework region to make the protein more stable. Actually, this is the case in the nature, and 40 years ago, David Baltimore lab and Who's lab, they found there's a higher hypermutation frequency at the CDR compared to framework. In the following works, many people in the field have shown this hypermutable CDR is not caused by cellular selection. It was an intrinsic pattern of this mutation. That's why in most of the immunology textbooks, people always draw more mutation at the CDR compared to framework. However, the molecular mechanism is still unknown. So our question is how this CDR regional hypermutability is achieved, and more directly, why the same WRC motif, whenever they are located in CDR or framework, they have different mutabilities. We think this mystery could be lying to a mesoscale that is 10 to 50 base per nanometer scale. So we started using biochemistry to study this question. We combined classic biochemistry, DNA assay, and next generation sequencing, which method allow us to check the biochemistry on more complex substrate. For example, on this framework CDR endogenous substrate, in the test tube, we found actually CDR also show a hypermutability. So it seems this hypermutability is all determined by protein and its substrate. And next, we extended our biochemistry to nearly all the known IgV genes, which including a substrate from 27 species and more than 1,000 V gene segment. And in this sequence, we observed similar things. That is, the CDR region is hypermutable in vitro. So this directly suggests the DNA coding sequence actually is the primary cause of this hot CDR phenotype, but not transcription, prompting, or prompting tag domain, something else. So if we consider the WRC feature as a mesoscale feature of ANE deamination, we wonder whether at the mesoscale, whether the surrounding sequence could play a hero support function to support the mutability of the WRC motif in their neighbor. So we started to investigate this question with different kind of mouse models. And in this mouse model, we knock in a scramble sequence and antibody gene coding sequence. And in the scramble sequence, we kept 6 AGCT at their endogenous positions, but scramble the surrounding sequence. And here you can see the CDR preference is almost gone here. Then next, we developed a faster way to generate mouse models with a mesoscale sequence alteration. And with this bunch of mouse models, we can alterate the surrounding sequence of this AGCT in CDR3, but kept CDR1 and 2 intact, which can serve as an internal control for control or most of the experimental variables. And we found actually the local sequence can affect AGCT mutation. And furthermore, if the sequence alteration is more close to AGCTA, have a stronger effect. Next, we want to exclude the mutation frequency is caused by downstream DNA repair pathways. So we knock out UG mismatch in B cell lines, and we check the authentic AID footprint here, and we show the similar observations. So that is in vivo, the adjacent sequence did affect AID activity. So the next question is how AID sense this information. We started with biochemistry with this mononucleotide contact. We put AGCT in this poly-DT or poly-DA contact. And we found AID actually prefer deaminate AGCT in this poly-DT contact. And the secret actually line into, at the protein side, it line into the positively charged surface. And as this critical positive charged amino acid are very important for this deamination in vitro, and AID actually binding this poly-DT substrate much better compared to this poly-DA sequence. So there is a preference of binding to as the protein and its substrate. So we wonder whether what it is a specific binding or it's a non-specific binding, or there's any sequence feature of this AID preferred mesoscale sequence. We started this from MD simulation because when they solve the protein structures, the substrate is not cannot be visualized. We found that when in the MD simulation, we found poly-DT sequence actually have a higher tendency to wrap around on the surface of this small protein, and this is correlate with the previously measured flexibility of the poly-DT and the poly-DA, and the T sequence is more flexible, A is more rigid. And furthermore, we can play around with the sequence. We can insert A6 or just the poly-DA hexamer in different position of these primers, and we check the activities by high sequence, and we found there is a positive correlation between flexibility and AID activity. So I think there is a correlation, and if you manipulate the DNA sequence, it can cause AID activity changes by changing the single-strand DNA substrate flexibility. Next, we wonder what kind of motifs are enriched in this flexible sequence. Many years ago, people have measured this dinucleotide stacking free energies, and they found pyrimidine dimers actually are more flexible. And here we show a very nice correlation between the flexibility and mutability. And if you put more pyrimidine dimers in this substrate, it have a higher deamination activity. Then we move on to the trinucleotide analysis. We found the antibody sequence. If we compare CDR framework sequence, actually there are two levels of selections. The first is at the amino acid level, and the second is at the DNA sequence level, which suggests that actually the antibody gene sequence is the coding sequence actually have non-coding roles. What can we do with these features? Here we show if we insert a flexible DNA sequence in cell lines in this CDR or code framework, we can turn them into a hotspot. And the same thing can be done in mouse models. We can make the hot framework into the code framework 3 become hot regions. So our work, we think we uncover a mystery feature of how this CDR hypermotility is achieved. We think the antibody coding sequence actually have evolved these hyper flexibility mesoscale features to support its hypermutability in antibody somatic hypermutation. And actually, this is not only observed in antibody coding sequence, And because AID can occasionally target too many proto-oncogenes to produce translocations, we found as this AID off target. We also found this correlation between flexibility and somatic mutation frequency. Okay, I think I will stop here by thank people in my lab who did this work. Yan Yan, who is at the far left of this in this photo, she did most of this work, and we have a lot of collaborators who contributed in these works. Most of the mouse models was from Dr. Yi and from Dr. Ott lab. And Lin Tai performed the molecular dynamic simulation for us. And we have mathematicians who help us to establish math models and check all our statistics. And Dr. Zhao helped us to analyze the IGV sequence from an evolutionary aspect. We also have a team to help us set up single molecule studies in this work. And Dr. Chao helped us to analyze a lot of informatics sequence features. And we have an enzymology expert, Xiaolong, on board for us to check all these enzyme dynamics. And I also want to thank the funding agents for funding us, and thanks for the attention. I'm happy to take questions. Thank you.
Thank you. Any questions? First, we can ask if any questions from online. So, on site.
I'm not very familiar with the cell culture model. Do you actually express AID in a B cell line, and you can continuously mutate the antibody genes without selection?
The answer is you can overexpress it, but you cannot properly control where to target. So basically, the antibody gene specific targeting is only we can only recapitulate this in mouse models. So for the variable regions, for hypermutation, in the B cell lines, you see some accumulations, mutation, even you overexpress AID on the IGV, the variable regions, but the efficiency is so low, it's never comparable to the mouse models. That's why a lot of our experiments we have to use mouse models to check. Otherwise, by some unknown reasons, this cannot be recapitulated in cell lines.
But in mouse model, I think you probably you do not immunize the mouse, or do you immunize the mouse? If you don't, why you actually see somatic hypermutation happening?
Because the mouse it take food, right?
I see.
It
Okay.
Food, there's microbiota. So
Okay.
whenever you check the B cell, they always immunogen or antigens.
Yeah, but this is difficult then to say, okay, the hypermutation hotspot is not because of selection. It's it's difficult to really rule out this possibility.
Yeah, to prove it, we use a non-functional allele. So
Okay.
basically, we have one allele coding antibody, another allele just for a test. So we have to do this on this non-functional allele. Yeah.
I see. Okay, right.
So, very great talk, Dr. Meng. This is Zhou Bing from Shenzhen Bay Lab. I want to ask a question about the proposed model of the AID hypermutation that is likely from your talk that are caused by the surrounding sequence that are one of the more major determinants for how to determine the CDR as the hypermutation regions. So if that is the case, then the model will suggest the AID will have ability by a cell or by another protein that can denature the DNA into single-stranded on the V area, the regions, as well as maybe a scanning mechanism of the enzyme to be able to scan through the surrounding sequence, determine where is the CDR. So any thought on this particular purpose on the enzyme property?
Oh yeah, AID cannot unlike the proteins, so there are a bunch of factors helping. Actually, it's targeted to the main target of AID is always transcribe. So transcription open the double-strand DNA. Then there's a lot of other factors, RPA, also RNA exosome. We showed it many years ago. RNA exosome can help AID. Can maintain this single-strand substrate for AID, but for the work I just presented is at the mesoscale level. Whenever we have this transcription bubble, that's how it determined on this scale. So,
any property of the enzyme they're able to scan the regions?
I think a lot of people many years ago have tried to check the precisity of the AID. I think for this situation, we more prefer a hit and run model for how this enzyme works. So it hit if it's not flexible, it's too rigid. Probably it binding as a K off. Maybe it's decrease or increase. Or then whenever you have a more flexible substrate, it lighter substrate, it binding longer, or then it have a higher chance to generate mutations.
Okay, thanks.
Okay, and thanks, Professor Fino again. And next, we move to next.
Okay,
thank you. And now we move to next speaker. The next speaker is Professor Lei Shi. He is from Tianjin Medical University, and today he will talk about the ADAR1 links R-loop resolution to ATR activation in replication stress response. Welcome.
So first of all, I want to thank the organizers for giving me the opportunity to present my lab's work. Here I'm going to talk about ADAR10 in R-loop homeostasis and ATR kinase activation. In dividing cells, the frequent occurrence of replication stress is a major source of genome instability. The stored or collapsed replication forks are marked by an accumulation of single-stranded DNA that are coated by replication protein A. When the actively transcribing RNA polymerase encounters with the replication fork on the same DNA template, the nascent transcript may reanneal with the template DNA, leaving the non-coding strand unpaired and forming a structure named R-loop that may threaten genome integrity. Protein kinase ATR is a master regulator in responding to replication stress. ATR forms a stable protein complex with its obligatory partner A-trip. Once the ATR A-trip complex is recruited to RPA single-stranded DNA template form, ATR could be fully activated by TAU-BP1. The recruitment of TAU-BP1 to blocked forks requires the expanding to Rad9 of the 911 complex. Then ATR phosphorylates RPA2, CHK1, and other effectors to protect fork and orchestrate cell cycle progression. As we talked about earlier, R-loop is composed of DNA hybrid, a stretch of displaced single-stranded DNA, and a trailing single-stranded RNA overcome. Tight regulation of R-loop generation and resolution is of utmost importance to maintain the physiological roles of R-loop, including class switch recombination, DNA repair, and the transcription termination, and to avoid genome instability associated diseases, including tumorigenesis as well as neurodegeneration. Until now, A plethora of factors have been identified for cells to counteract harmful envelope accumulation. These factors include RNase that degrades the RNA hybridized with DNA, a number of helicases, RNA processing factors, DNA repair factors, and chromatin modulators, as I listed here. Adenosine to inosine editing of RNA is a prevalent type of RNA modification. ADA1 acts as a ubiquitously expressed RNA editing enzyme, and it exists in two different isoforms. A constitutively expressed 110 kilodalton nuclear protein and the interferon inducible 150 kilodalton cytosolic protein. They share a copy of an N-terminal ZDA binding domain, 3 repeated copies of a double-stranded RNA binding domain, and a single deaminase domain in the C-terminus. Mounting evidence proves that ADA1 plays a pivotal role in maintaining self-tolerance and preventing autoimmunity through editing double-stranded RNAs in the cytoplasm. Otherwise, the unedited double-stranded RNA will activate innate immune sensors MD5 and PKR. ADA1 could also function independent of its editing activity by forming regulatory complexes with RNAs or proteins. For example, ADR could coordinate with DICER to promote microRNA processing. ADA1 is able to antagonize START1 mediated messenger RNA decay by competitive binding to the 3' UTR of double-stranded RNAs. Erroneous response to replication stress is believed to be a potent driver of functional loss in hematopoietic stem and progenitor cells that play a key role in hematopoietic reconstitution. Our collaborators, Professor Tao Chen and the other groups, have reported that ADA1 deficient mice display the rapid induction of apoptosis in hematopoietic cells, and ADA1 loss causes DNA damage and sensitizes tumors to irradiation. This evidence triggered us to hypothesize that ADA1 is involved in replication stress response. To test this idea, we generated ADA1 knockout HSPCs and confirmed that ADA1 deletion significantly inhibited the colony formation of these cells. Then we performed cognitive assay and immunostainings. The results here show that ADA1 loss was associated with increased DNA tail movement and high levels of gamma H2AX in EDO-positive replicating cells. We next generated multiple ADA1 variants, including P150, P110, and double-stranded RNA binding defective mutant EAA, deaminase that mutant of P110. Rescue experiments, as manifested by gamma H2AX staining and common tail evaluation, show that only P150 and the EA mutant that can bind double-stranded RNA fail to efficiently suppress ADA1 knockdown-induced DNA damage accumulation. Once ADA1 and MD5 or PKR were co-depleted, we can see that it couldn't neutralize ADA1 ablation-induced effects. This data raised the possibility that the double-stranded RNA binding domain unknown the catalytic activity of ADA1 P110 is indispensable for genome integrity. To understand how ADA1 controls genome stability, affinity purification and mass spectrometry were employed to identify the interactomes of ADA1. The results indicated that TALBP1 was one of the top candidates. By means of co-immunoprecipitation assay, we found that the double-stranded RNA binding defective mutant ADA1 EAA was not able to interact with TALBP1, and the N-terminal BRCT from 0 to 2 is responsible for TALBP1 association with ADA1. The essentiality of these binding elements was further confirmed by GST pull-down experiments with recombinant proteins. These results suggest that ADA1 interacts directly with TALBP1. To assess that whether ADA1 plays a role in ATR activation, HSPCs were cultured in the presence of the topoisomerase inhibitor camptothecin to arrest the replication fault. Immunoblotting analysis indicated that ADA1 knockout led to vastly decreased phosphorylation level of CHK1 and RPA2, while rescue experiments showed that only ADA1 EA mutant that failed to interact with TOP1 is not able to revert ATR's kinase activity in ADA1 depleted cells. Thereby, we propose that the genome protection role of ADA1 entails its physical interaction with TOP1 rather than the AT1 editing activity. We next ask that whether ADA1 is required for the recruitment of TOP1 to replication stress sites. First, we show that ADA1 knockdown led to a significant decrease of TOP1 protein formation at CPT-induced replication stress sites, and this could be reverted by enzyme guide ADA1 but not EA mutant. We next found that ADA1 loss weakened the interaction of Reg9 with TOP1, while EA mutant couldn't restore the binding defect. According to these observations, we propose that ADA1 could orchestrate the loading of TOP1 and damage the replication forks, thus promoting ATR kinase activity. Considering that ADA1 loss-induced DNA damage predominantly occurs in S phase of the cell cycle, and R loops are major source of replication stress and genome instability, we wonder that whether ADA1 is involved in R loop homeostasis. With proximity ligation assay, we show that the RBDs of ADA1 as well as the key residues in double-stranded RNA binding are responsible for ADA1 interaction with DNA hybrids. The importance of RBDs is further confirmed by jump-shift assays. We then performed strip-seq experiment and show that ADA1 deficient cells bear higher levels of R loop at the gene body, promoter, and downstream region. Well, RNase-H1 treatment efficiently degraded DNA hybrids at R loop 4 regions along the genome. This R loop unwinding factors DHX9 and DDX21 could be also co-purified with ADA1. We wonder that whether ADA1 may rely on these helicases to resolve R loops. Here we can see that ADA1 depletion significantly impaired the PLA signals between DNA hybrid and the DHX9 of DDX21. Well, this effect could be restored by overexpression of wild-type and enzyme-guide ADA1, but not DAA or RBD-like mutant. According to this data, we propose that ADA1 suppresses the R loop formation through physically engaging RNA helicases DHX9 and DDX21. Replication errors or defects may block the progression of transcription apparatus and generate genome destabilizing R loops. Thereby, we ask that whether ADA1 promoted R loop resolution is functionally linked with ATR activation in the context of replication stress. The ProIP data shows that hydroxyurea treatment resulted in a marked reduction of ADA1 TOP1 binding, while the interaction between ADA1 and DHX9 or DDX21 was strengthened. Importantly, we noticed that this operation could be reverted by either DRB treatment or forced expression of RNase-H1. This data imply that ADA1 mutated protein complexes are dynamically regulated, and R loops play a role of importance in controlling ADA1 redistribution in responding to replication stress. Indeed, we found that DNA hybrids addition impaired TOP1 ADA1 binding in a dose-dependent manner. Moreover, we revealed that comparing to enzyme-dead RNase-H1, overexpression of this wild-type compound led to an enhanced colocalization of ADA1 and TOP1 at the replication stress sites, and weakened the ADA1 signal at R loop regions. These results suggest that R loops raised by replication stress with crosstalk way and actively extract ADA1 from perturbed replication faults. To address the biological significance of ADA1 redistribution, we examined that whether our loops contribute to ADR activation and perturbed replication faults. From the immunostaining data we showed here, we can clearly see that DRB treatment of RNSH1 overexpression will significantly reduce ATR activity. We then introduced ADA1 into HeLa cells and examined the phosphorylation level of our PA2 and CHK1 and found that the ATR kinase activity was increased in and then dead RNSH1 expressing cell, but not wild-type RNSH1 expressing cell, suggesting that our loop will facilitate ADA1 redistribution to promote ATR activation. At the end, I would like to summarize the key points of the story. Briefly, we found that the nuclear expressing ADA1 plays a critical role in genome integrity through promoting ATR activation and genome-wide R homeostasis. And the genome protection role of ADA1 heavily relies on its double-studded RNA binding domains, but not the catalytic activity. When replication is inhibited, RNA hybrid competes with POP1 for ADA1 binding to promote the translocation of ADA1 from damaged fork to R-loop region. ADA1 then recruits RNA helices for R-loop unwinding and simultaneously allowing POP1 to more efficiently stimulate ATR. The take-home message is that R-loop will crosstalk with blocked replication fault, at least by transposing ADA1 to fine-tune ATR activity and safeguard the genome integrity. To conclude, I would like to thank my lab members Shuai, Bill, Adi, and our collaborators, Hao and Hui. Also, I want to thank the funding agencies that support our research. I would like to stop here and thank you very much for your attention.
So, thanks, Professor Shilei. And any questions? So first, give a chance for online. Any questions for online? No? Oh, on site.
I'm interested in the Delta CD variant. So it seems the Delta CD variant somehow couldn't function to activate ATR, but it can still be recruited to the R loop. Am I correct?
Actually, in our data, we showed the ADA1 lacking the catalytic domain could also activate the ATR, and it could also bind the DNA hybrid.
So what's the function of the catalytic domain?
The catalytic domain functions to deaminate the adenosine, but in our story, I think it is not involved in ATR activation and R-loop homeostasis.
Okay.
Thank you.
Hello, can you hear me?
Sure.
Very nice talk, and I have a very simple question. So I remember ADAR1 has two isoforms. One is the P110 and 150. So which one do you think is the one more important, or both are required?
In our model, I think the P150 it plays a major role, and the P110 it predominantly localized in the nuclear, and it should be involved in the genome stability.
So I also wonder, for these two isotopes, are there like a cell type specific expression, or both are expressed in universal?
I think the P110 should play a universal role.
Okay, thanks.
Thank you.
Okay, thanks, Professor Shilei again. And now we move to next speakers. Thanks.
Thank you.
Okay, goodbye. And the next speaker is Dr. Zhiyong Mao. He is the professor and the director from Clinical and Translational Research Center of Shanghai First Maternity Infant Hospital, Tongji University. And today he will talk about the establishment and application of the reporter system for DNA repair analysis. And welcome.
First of all, thank Xinzhi for the invitation. It's really a great honor to be invited to give this talk. Our lab is mainly on DNA repair and aging. So today I will summarize what we did with the tools for analyzing DNA double-strand repair and tell some stories about its utilization. The first part is about the tools. I believe that everyone in the audience is familiar with the two repair pathways for fixing DNA double-strand breaks. One is NHEJ, the other is HR. The defects in the two repair pathways will lead to the rise in genomic instability, causing tumorigenesis or the premature aging. Most of us used biochemical assay, common assay, or counting foci in analyzing DSP repair efficiency, but these methods are usually quite laborious, time-consuming, not sensitive, or precise enough. So people in this field have created a number of fluorescent protein-based DNA repair measurement systems, which can rapidly, precisely, and quantitatively analyze the efficiency of DSP repair. Our group has also made some contributions to generating the repair cassettes. During my PhD and postdoc, we created the NHVJ and HR reporters, and using these tools, we tried to solve a number of aging-related problems. I studied my own lab. We asked whether it is possible to build a reporter that can simultaneously measure the efficiency of NHVJ and HR at the same chromosome size. We successfully made the reporter based on GLP and TD tomato, and the cell lines that have the reporter integrated into the genomes. NHVJ-mediated repair will turn cells green, while HR will turn cells red. We can assess them on FACS or using confocal microscopy. We also validated this reporter system by checking the balance in different cell cycle stages or knocking down some critical NHVJ or HR factors. These data indicate that we successfully obtained a system that can measure both HR and NHVJ at the same time. But we have to introduce the exogenous IC1 expression vector into the cell lines, which is also a little annoying. So we decided to build another doxycycline-inducible IC1 vector and integrated this vector into the genomes of the reporter line and created this so-called CLZ3 cell line. With the supplemented dots to the cell line, IC1 would start to express, not just the recognition size, and we can start to measure the repair efficiency either using FACS or microscopy. Using this system, we did the large-scale screening on a number of small molecule chemicals and obtained some chemicals like Ferrara, which can promote HR but have no effect on NHVJ. Using Ferrara, we demonstrated that it can be applied to improve genome targeting efficiency. These studies were based on reporter cell line. Is it possible to measure the efficiency in vivo? Again, during my PhD, I modified the NHVJ reporter and integrated the reporter into those 26 sites of mouse genomes and successfully made the NHVJ mice. Afterwards, in my own group, using the same strategy, I also obtained the HR reporter mice. Using the two reporter mouse models with the hydrodynamic tail vein injection method, we introduced the IC1 vectors to the liver and measured the efficiency of both pathways in the liver. We found that both HR and NHG were significantly elevated in hepatocellular carcinoma in comparison to adjacent normal liver tissues, suggesting that targeting the two pathways may be an effective approach to treat HCC. In the previous work, we mentioned that we created the dual fluorescence reporter for measuring both HR and NHG at the same chromosome site. So is it possible to use the same strategy to create the reporter mice for measuring both HR and NHG in different tissues or organs? With this idea, we modified the two constructs and successfully created the dual fluorescence reporter mice. We also integrated the DOX-inducible IC1 construct into H11 site and obtained the DOX-inducible IC1 mice. We bred the two mouse models and fed the crossed line with doxycycline. We successfully observed P or TD tomato positive cells, which reflect NHG or HR events in different organs. So with this in vivo model, is it possible to link the impairment in DNA repair in tissues or organs to aging or aging-related diseases? With this question, we studied progeria, in which Professor Baohua Liu has done wonderful work. Progeria patients have very special phenotypes from cells to individuals. They often die from heart complications. At the molecular level, the cause of the progeria is that there is a synonymous mutation at G609 site, although this mutation does not cause a change in amino acids. But it changes the splicing, which leads to the loss of 50 amino acids. The lost 50 amino acids contain a site that can be cut by ZMPSTE24 enzyme. So it will lead to the accumulation of the so-called progerin and loss of the mature lamin A. Although we know that the genomes in HTTPS patients or mice are destabilized, the regulation of NHG has not been well characterized. Some reports are even contradictory. We speculated that the regulation of NHG might be context dependent, whether there is a change in NHG in hearts, particularly in cardiomyocytes, and the potential regulatory mechanisms remain to be determined. The major reason is that there is no tool for studying DNA repair in cardiomyocytes, and isolating, culturing cardiomyocytes, or introducing exogenous genes into cardiomyocytes is technically challenging. We first counted the gamma-H2X foci and looked at the genomic stability using common assay in HTTPS cardiomyocytes. We confirmed that gamma-H2X level was elevated, and genomes were more unstable in HTTPS cardiomyocytes than in normal cardiomyocytes. Next, we bred our reporter mice with doxycycline-inducible IC1 mice and examined the NHG efficiency in hearts. After feeding these mice with doxycycline supplemented water, we examined the IC1 expression in hearts using immunostaining assay. Interestingly, we found that IC1 happened to express only in cardiomyocytes, but not cardiac fibroblasts. We successfully observed GP positive cells, which reflect NHG efficiency. We found that in normal hearts, 30% cardiomyocytes turned green. This data demonstrated that this system could be used to analyze NHG efficiency in cardiomyocytes. We collaborated with Professor Baohua Liu and obtained the HTTPS mice. By crossing these mice with our reporter mice, we also examined the NHG efficiency in HTTPS hearts. We found NHG declined by 50% in HTTPS hearts in comparison to in normal cardiomyocytes. What causes the decline in NHG? We found that in comparison to wild-type lamin A, progerin cannot be recruited to DSP cells. The reason is that lamin A can interact with gamma-H2X, while progerin loses its ability to interact with gamma-H2X. The further study demonstrated that both the missing 50 amino acids and the phosphorylation modification contributed to the attenuation of gamma-H2X progerin interaction. The previous work by Professor Zhongjuan Zhou and Baohua Liu indicated that lamin A is the endogenous activator of 36, which initiates NHJ signaling cascade. Our further studies supported their finding too. We found that in progeria hearts, 53BP1 recruitment was also impaired. During the analysis of NHJ efficiency and mechanistic study, we observed a weird phenotype. That is, it seems that the NHJ mice hearts are relatively small. We confirmed that the ratio of the HTTPS hearts versus tibial length is lower than the ratio of wild type. And we also observed reduced size of the cardiomyocytes in HTTPS mice. These data suggest that HTTPS mice have a phenotype of cardiac atrophy. Consistently, we also observed reduced heart functions in HTTPS mice. Then we asked whether there is a mechanistic link between dysfunctional NHJ pathway and the phenotype of cardiac atrophy. To answer this question, we examined whether the decline in NHJ will lead to the changes in the expression of post-translational modifications of DDR factors. Indeed, we found that the expression level and the phosphorylation level of CHEK2 were upregulated in HTTPS mouse hearts. And we also observed the phosphorylation level of AMPK alpha, which is critical to the regulation of cell size, was upregulated in HTTPS hearts. So we wondered if CHEK2 as a kinase participates in the phosphorylation of AMPK alpha. We performed CoIP and PLA assay, and we found that there was interaction between CHEK2 and AMPK alpha in the nucleus. Further experiments indicated that CHEK2 took part in the regulation of AMPK alpha phosphorylation, but the enzymatically dead CHEK2 mutant or CHEK2 inhibitors did not change the CHEK2-mediated upregulation in AMPK alpha phosphorylation, indicating that this regulation is independent of its enzymatic activity. The known upstream kinase of AMPK is LKB1, so we hypothesized that CHEK2 might promote the LKB1-AMPK interaction, thereby promoting its phosphorylation. Our further CoIP experiments indicated that this is true. Overexpressing CHEK2 promoted their interaction. Once AMPK alpha is phosphorylated and activated, it will phosphorylate FOXP3A, promoting the expression of essential genes involved in regulating cell size. And our experiments indicated that FOXP3A phosphorylation level was increased in HTTPS hearts, and its downstream genes were upregulated too. Based on the observed phenotype of cardiac atrophy, we asked whether forced enlargement of the hearts could extend the lifespan of HTTPS mice. We used the widely used isoproterenol to treat HTTPS mice. We found that after the treatment, the sizes of HTTPS hearts were rescued. And this treatment suppressed the AMPK FOXP3A signaling axis, but it did not affect its genomic stability. The heart functions were also rescued. Importantly, both the mean and maximum lifespan was significantly increased by 17%. Overall, we created the novel reporter mice and analyzed NHJ efficiency in HTTPS mouse hearts and found that NHJ declined in cardiomyocytes. This decline was caused by the impaired interaction between progerin and gamma-H2X, leading to failed recruitment of progerin to DNA dendritocyte. Persistent DDR response promoted the HR protein level, stimulating the LTB1-MPK interaction. The activated MPK reduced the size of cardiomyocytes, causing the phenotype of cardiac atrophy and impaired heart functions. This work was mainly done by Dr. Yu Chen and a PhD student, Shiqi Huang. Also thank all my collaborators, Professor Ku Wei, Professor Baohua Liu, and Professor Vera Govanova. Thank all the funding agencies for giving us money to finish this work. With this, I will stop here. And thank you all again.
Okay. Thanks, Professor Jim. And any questions? So first, give chance for a person online. Any questions from online? Still no? Okay, on site. Wow.
So there's first the basic point I want to make sure I understand. So for your progerian mice, do you inject plasmids to somehow put the plasmids into heart, or you actually generate a reporter-bearing mouse?
We generate, yeah, that's a good question. Yeah, we generate the reporter mice and bred with HPS mice. So it's quite time consuming. Yeah, that's what we did. Yeah.
So for the progerian, because of the involvement of laminin, all this, does this mean the NHEJ repair happen at the nuclear lamina, or somehow the laminin actually go to the nucleoplasm to do the DNA functional DNA repair role?
Yeah, I think that is what Baohua and Zhongjin did before, and some other labs also did that. Yeah, laminin. From our data, yeah, laminin would.
Yeah, sure, but the process happened at nuclear periphery, the nuclear lamina region, or somehow.
Yeah, that good question. We don't know.
So because I wonder if because you make this mouse, so what side do you integrate? Do you integrate into the nuclear lamina associated domain, or actually you integrate into a interlamina domain? Therefore, actually, where do they find?
We should check. We integrate our reporter into LOSA 26 side. We don't know whether this side is close or is just located to the laminin A control region. We don't know. We need to check. That's a very good point. Yes, thanks.
Okay, and then Professor Zheng, please check the question on Tengxun meeting, and the question has been typed. You can check it.
Oh, I did not open that. I only have the Zoom.
Okay.
Oh, yeah, I have a question, so now I can speak. So how does Zheng, great work and Feng here, from SUSTEC. So I'm curious, how does isoproterenol actually stimulates the activity of? Is it activating the LKB1 AMPK pathway while directly acting on CHEK2, or is through some other mechanisms?
Yeah, that's a good point. I guess there is no direct evidence showing this ISO can directly target NPK or something. I think it just forces the cardiomyocytes to grow larger. Okay, and yeah, we're trying to inhibit NPK right now, and to see if this assay can also promote the size of hearts and extend lifespan of HPS mice. Yeah, that's a good point. Yeah.
Okay, thank you.
Okay, any questions? No.
Thank you so much for your talk.
Okay.
I have two questions. One is,
Alright.
have you seen any loss of heterochromatin because you are knocking out laminin A?
The loss of heterochromatin.
Heterochromatin, especially in laminin-associated domains. I mean, when you are studying, have you seen some epigenetic changes, especially loss of heterochromatin in your knockout mice?
In that knocking mice, I think we did not check that. We just try to connect deficiency in DNA repair to the phenotype cardiac atrophy. At the molecular level, we did not check that, but I guess someone else has already done this before in some other published work.
So another thing, inhibition of FOXO3A gene, right? So FOXO3 basically is a aging gene which promotes the aging or longevity, and it is strong antioxidant, have capabilities to antagonize the oxidative stress. So when you just inhibit that activity, it must have some adverse effect on aging. So have you compared that if we can bypass this inhibition and compare the results with so what is the efficiency? I mean, if we just bypass FOXO3.
Yeah, that is also a good point, I think. Yeah, FOXO3 is quite important. I mean, in the activation is quite important to inhibit apoptosis and promote cell survival, and it's also related to longevity. I guess it's in different biological contexts, it plays different roles. I think in the context of cardiomyocytes, persistent activation of FOXO3A may force cells to grow smaller, and which can lead to this cardiac atrophy phenotype. So, yeah, it is a good point. I think at this point, at this level, yeah, it probably plays some adverse rules.
Thank you.
Because of the time, we had to stop and move on to next speaker. And thanks again, and very nice work.
Thank you.
Okay.
Thank you.
Thank you. And the next speaker is Professor Qiang Chen from Wuhan University. He will talk about SCML to inhibit DNA damage-induced cell death through regulating p53 and CHK1 stability. Welcome.
I want to thank the organizer for giving me this opportunity to report our research on DNA damage response. We identified SML2 protein as a novel regulator of DNA damage response, which could inhibit DNA damage-induced cell death through regulating p53 and CHK1 stability. DNA damage leads to the activation of protein kinase ATM and ATR. ATM could activate p53, which upregulate the transcription of genes involved in apoptosis or cell cycle arrest. CHK1 could also be activated after DNA damage, which induces the activation of SG2 or G2M checkpoint. This cell cycle arrest induced by p53 or CHK1 activation may reduce the accumulation of DNA damage in cancer cells and help cancer cell escape from DNA damage-induced cell death. We found that SML2 participate in both p53 and CHK1 pathway after DNA damage. In mouse, SML2 mainly expressed in testes and functions in spermatogenesis. SML2 could interact with USP7 and modulate S2A monoubiquitination in spermatogenesis. However, SML2 has two isoforms in human. The similarity between mouse and human SML2 is low. Previous studies have reported SML2 could stabilize PRC1 complex and regulate histone ubiquitination, and SML2B could stabilize P21 through direct interaction, but the role of SML2 in DNA damage response is still unknown. To examine the role of SML2 in DNA damage response, we firstly studied the effect of SML2 depletion on cell survival after DNA damage. SML2 depletion in HCT116 cells showed significant decrease of cell survival after CPT or HU treatment. The same result was observed in other two different cell lines. These results suggested that SML2 may contribute to the resistance of cancer cells to DNA damage. To explore the role of SML2 on DIN damage response, we firstly examined the effect of DIN damage on SML2. Both IR and CPT treatment obviously increased SML2 protein level. The mRNA level of SML2 was intact after CPT treatment, suggested that the protein stability of SML2 may be upregulated after DIN damage. To confirm the increase of SML2 stability after DIN damage, the half-life of SML2 was measured after CSH treatment, which was a protein translocation inhibitor. We found DIN damage increased the half-life of SML2. Thus, we uncovered the effect of DIN damage on SML2, which led to SML2 stabilization. As protein phosphorylation is widely involved in DIN damage response, we want to know whether SML2 could be phosphorylated after DIN damage. We found that SML2 was phosphorylated after CPT treatment, as plotted with phosphoantibodies. This modification was inhibited by ATR or CHK1 inhibitor treatment, but not ATM or DNPK inhibitor, indicating CPT-induced SML2 phosphorylation was mediated by ATR-CHK1 pathway. We identified 570 serine of SML2 was the phosphorylation site as S578 mutant was weakly phosphorylated after DIN damage. Furthermore, using in vitro kinase assay, we found CHK1 could directly phosphorylate SML2. This result suggested that CHK1 was responsible for DIN damage-induced SML2 phosphorylation. As DIN damage increased the stability of SML2, we compared the half-life of SML2 and found that S570E mutant, which mimic the DIN damage-induced phosphorylation of SML2, exhibit greatly increased stability. Thus, we discovered that DIN damage-induced SML2 phosphorylation, which could increase SML2 stability. To study how SML2 was stabilized after phosphorylation, we did affinity purification using SML2 as a bait. We identified DDB1 as an intacting protein of SML2. DDB1 was a component of known E3 enzyme. Thus, we proposed that DDB1 mediated ubiquitination of SML2, which led to its degradation. DDB1 depletion increased SML2 protein level and reduced its ubiquitination, suggesting that DDB1 indeed mediated the degradation of SML2. Furthermore, S570E mutant decreased the interaction between SML2 and DDB1. Thus, we concluded that after DIN damage, CHK1 mediated S570 phosphorylation blocked the interaction between DDB1 and SML2, which led to SML2 stabilization. We have uncovered the effect of DIN damage on SML2. Next, we studied how SML2 regulates DIN damage response. Firstly, SML2 is a tighter USB7 intacting protein. We want to know whether this interaction was involved in DIN damage response. As how the interaction between USB7 and SML2 was mediated was still unknown. We mapped the interaction region of USB7 with SML2. We found that trap domain of USB7 was responsible for this interaction. Trap domain is a well-studied domain in USB7 which mediated its interaction with multiple proteins, including p53 MDM2. The binding motif of trap domain was PAXXS. Based on the known binding motif, we analyzed the protein sequence of SML2 and found 5 motifs. We constructed point mutant according to these motifs. Using in vitro binding assay, we found only PVNS motif was critical for the interaction between SML2 and USB7, as S441 mutant totally lost the interaction with USB7. The same result was observed when the mutant was expressed in cells. Thus, we identified the key residue for the interaction between SML2 and USB7. As trap domain of USB7 could interact with p53 and MDM2, we compared the binding affinity of USB7 with p53 MDM2 and SCML2 using MST assay. We found the binding affinity of SCML2 with UIP7 was similar with MDM2, but is higher than p53. Using the binding assay between UIP7 and p53, we found SCML2 wild-type protein could block the interaction between UIP7 and p53, but not SCML2 as 441A mutant. Thus, SCML2 could compete with p53 for the interaction with UIP7. As UIP7 could regulate p53 stability after DNA damage, we assume that SCML2 may participate in DNA damage response through affecting the binding between UIP7 and p53. We established dock-inducible Taiton cell line for SCML2 expression and found SCML2 overexpression decreased p53 protein level after DNA damage in a dosage-dependent manner. The expression of p53 downstream genes was also affected. For example, the apoptosis-related gene. The same effect was observed in ACT116 cells, where SCML2 was overexpressed. On the contrary, SCML2 depletion reduced the ubiquitination of p53 and increased the p53 protein level. This result suggested that SCML2 may regulate DNA damage response through affecting p53 stability. To confirm this hypothesis, we overexpressed wild-type SCML2 or S441 mutant in ACT116 cells and examined the cell survival after DNA damage. We found wild-type SCML2 greatly increased cell survival, but S441A mutant only has mild effect. Finally, we concluded that DNA damage-induced SCML2 stabilization could compete with p53 for its binding with UIP7, which led to the decrease of p53 protein and reduced p53-mediated cell death. What happened on p53 mutant or p53 non-cancer? We carefully examined the CXQ1 status in p53 negative cells after DNA damage. Interestingly, we found the extent of CXQ1 degradation after DNA damage was enhanced in SCML2 depletion cells, suggested SCML2 may protect CXQ1 from DNA damage-induced degradation. We also found the ubiquitination level of CXQ1 was increased after SCML2 depletion. CXQ1 could be degraded by several different pathways, such as protein-mediated degradation. As to DNA damage-induced degradation, two types of E3 was involved. Both of them targeted the C-terminal of CXQ1 for ubiquitination. We analyzed the region of CXQ1 responsible for this interaction with SCML2 and found C-terminal of CXQ1 intact with SCML2. We proposed that SCML2 may block the ubiquitination site recognized by E3 through its direct interaction with CXQ1. We expressed the C-terminal CXQ1 and analyzed its ubiquitination modification and found that SCML2 overexpression indeed reduced the ubiquitination of C-terminal CXQ1. This result suggested that SCML2 stabilized CXQ1 through its interaction with CXQ1 C-terminals. Whether SCML2-mediated CXQ1 stabilization contributed to the resistance to DNA damage, we treated SCML2 overexpressing cells with CXQ1 inhibitor and found that CXQ1 inhibitor treatment reduced SCML2-mediated resistance to CBT or HU treatment. In conclusion, we found in p53 negative cells, the interplay between CXQ1 and SCML2 leads to resistance to DNA damage treatment. To confirm this conclusion, we established chemo-resistant cells in p53 negative cells through two-step CBT treatment. We found in these chemo-resistant cells, SCML2 protein level was greatly improved. The stability of CXQ1 after CBT treatment was also increased in these cells compared to parental cells. Meanwhile, SCML2 depletion in this chemo-resistant cell significantly reduced the stability of CXQ1 after DNA damage. Furthermore, SCML2 depletion or CXQ1 inhibitor treatments increased the sensitivity of this chemo-resistant cells to CPT or HU treatment. Our result indicated that SML2 participated in DNA damage response. DNA damage induced CHK1-mediated SCML2 phosphorylation and stabilization. Functionally, SML2 may contribute to the chemo resistance of cancer cells via at least two mechanisms. First, in p53 positive cells, SML2 suppresses the stability and activation of p53 after DNA damage through competing with p53 for UIP7 binding. Second, in p53 negative cancer cells, elevated SML2 level after DNA damage stabilize CHK1, especially after DNA damage, which lead to the activation of CHK1 pathway to maintain SG2 or G2M checkpoint. This project was performed by two students. I also want to thank the following funding support. Thank you.
Thanks. And any questions from online? Any question? No, on site.
Have you knocked out or knocked down a USP7 in the knockout cell line to observe whether you rescue the phenotype?
Okay, this is a good question. Actually, we used the USP7 inhibitor to mimic the USP7 knockout, and we use the USP inhibitor. We think our series is okay. Yeah.
Another question. And very interesting work, and so I'm very curious that have you detected some mutation mutation on the point mutation that disrupt the interaction of SCML2 to detect the phenotype? Have you detected some mutation on SCML2 to the point mutation, or
you mean the cancer related?
No, the mutation that disrupt the interaction between SCML2 regulated pathway.
Mutation, yeah. Actually, yeah, we mutated the 441, and we just use this mutation just to want to study the whether H2 that SML2 worked with UIP7 to regulate the p53 pathway. Yeah.
Okay.
We didn't do other mutations. Yeah.
Okay. Thanks again.
Can I ask you one more question here?
Sure.
Hey, Qiang, great work, very interesting. And I see that you showed the DDB1 as the E3 ligase. So, but you also showed some decaps were actually put down in your mass spectrometry IP. Have you identified the exact decap for degrading SCML2?
No, we didn't do the further experiment to verify which one is actually the DCAF for SML2 degradations. The reason is we found that the SML2 phosphorylation could directly inhibit the DDP1 and SML2 interactions. We think that probably the DDP1 directly mediates the interaction, and the DCAF maybe only have the association. Yeah.
Okay, it's very interesting because some proteins, even they interact with the DDB1, they may not be the degradation target for.
Yes, you're right. Yeah. Yeah, very great suggestions. Based on our mass spec, I think we can to analyze in detail about which one is the exact one to mediate SML2 degradation or ubiquitination. Yeah, thank you.
And any other questions? So if no questions, we move on to next speaker. So thanks again. The next speaker is Dr. Yunpeng Feng. He is the associate professor in the key laboratory of molecular epigenetics of the Ministry of Education, Northeast Normal University. He is also my collaborator, and today he will talk about epigenetic mechanism on the activation of mammalian replication origins. Welcome.
Thank you, Haichen. Thank you. Can you see my screen? Can you hear me?
Yes, yes, we can see clearly and hear clearly.
Good, good. Thank you very much, Haichen. Special thanks to the organizers. Just need to get my pointer. There's a pointer. Yeah. It is a great pleasure for me to share our recent findings with you. Hopefully, I can convince you that the OGNAC modification on histone H4 747 can regulate DNA replication and is important for origin activation. It's not okay. Yeah, so in each round of the cell division cycle, eukaryotic cells have to make a decision. They can precisely duplicate the RD during propagation or experience programmed changes to achieve differentiation. But if unscheduled changes occur during this process, pathological situations, including cancer, can be promoted. Apparently, S phase is a very critical window, because during this specific period of time, the entire genome must be completely replicated. Eukaryotic DNA replication begins at various genomic locations, defined as replication origins, each of which gives rise to two replication forks traveling in opposite directions. The very first step in DNA replication is origin licensing, starting from the loading of OCC complex. Subsequently, CDT1 and CT6 are recruited to facilitate the loading of replicative helicase MCM complex. Once MCM complex are loaded, replication origins are licensed, are ready to be activated. Following MCM loading, CDC45 and genes arrive to activate MCM complex for DNA unwinding, which allows the progression of replication forks. Of note, in every cell cycle, eukaryotic cells have license excessive replication origins, but only a fraction of these origins can be activated to initiate DNA replication. How licensed origins are selectively activated still remains poorly understood. Although certain sequence features have been found in yeast, up to now, no consensus sequence that can comprehensively predict replication origins in mammals has been identified. In recent years, different factors, including chromatin configuration, histone modifications, have been found to be important for replication, for the control of replication origins. For instance, both acetylation and methylation of histone H4 have been found to be able to regulate origin activity. And beautiful work from Haichen showed that histone variant can also regulate origin activity by H4 K20 dimethylation. That's a beautiful work. And in 2010, H4 was also found to bear ognacylation, a unique type of post-translational modification recognized as nutrient sensor. Ognacylation usually takes place on serine or threonine and reversibly controlled by OGT and OGA. So far, only one ognacylation site has been identified on H4 747, but its biological function is still unknown. We are very much interested in post-translational modifications, including open-oscillation, ubiquination, and acetylation. We're wondering whether H4 747 open oscillation can play a role in DNA replication. We started with mutating S47 into alanine, which can't be open oscillated. But as expected, a dramatic reduction was detected upon S47 mutation, boosting open oscillation by OGA inhibitor PUG-NAC remarkably enhanced the open NAC modification level in H4 wild type, but not in the mutant, confirming S47 as the key open oscillation site on H4. To investigate the role of H4 S47 open oscillation in DNA replication, we labeled replicating DNA with EDU and marked S phase cells with PCA. Upon S47 mutation, DNA replication was repressed and PUG-induced upregulation was also compromised. Using an alternative approach, we confirmed the important role for H4 S47 open oscillation in DNA replication. To further differentiate the replication defect in initiation and elongation, we performed the DNA fiber assay under different conditions. Fork velocity remained rather stable. In contrast, upon S47 mutation, the origin density was reduced, and PUG-induced upregulation origin density was also abrogated, indicating that H4 S47 open oscillation is important for replication initiation. Meanwhile, we observed that OGT tended to enrich on chromatin in S phase cells and at least in partial resides at replication sites marked by PCA. More importantly, once S47 was mutated, the OGT-induced upregulation in origin density was blocked, indicating that OGT regulates replication initiation via H4 S47 open oscillation. Next, the most important question is how could H4 S47 open oscillation regulate DNA replication? Using mass spec, we detected a panel of candidate proteins which chose H4 wild type over H4 mutant. The presence of MCM246, which are different members of the replicated helicase MCM complex, immediately caught our attention. In the following validation experiment, we not only confirmed their association, but also found that the rest of the MCM complex could also preferentially bind to H4 wild type. And upregulation of open oscillation by PUG have further enhanced MCM association with H4 wild type, but not with H4 mutant. So as I mentioned earlier, replication initiation consists of two steps: origin licensing and origin activation, which can be reflected by MCM loading on chromatin and phosphorylation respectively. We first examined MCM loading on chromatin and found that none of the MCM members was changed upon S47 mutation, regardless of whether PUG or exogenous OGT was provided, suggesting that H4 S47 open oscillation may be not required for origin licensing. Therefore, we shifted our focus to origin activation, where the phosphorylation for MCM complex, for instance, MCM2 phosphorylation at S53, is often used as a proxy. Interestingly, once S47 was mutated, MCM2 phosphorylation was reduced, and PUG-induced upregulation was also impaired, indicating that H4 S47 open oscillation might be important for origin activation. Because CDK and DDK are master kinases, which control replication initiation, by catalyzing the phosphorylation of key components in the initiation complex, including MCMs. Neither in our experiment, neither H4 S47 mutation nor changes in open isolation altered CDK2 binding on chromatin. In contrast, the chromatin bound of DDK, including its catalytic subunit CDC7 and regulatory subunit DBN4, was reduced evidently. To further dissect the regulation of DDK complex by H4 S47 open isolation, we purified different types of H4 and DDK from E. coli. And we found that only the H4 wild type, but not the mutant, could interact with DBF4 and enhancing H4 open isolation by OGT can further improve this interaction. In contrast, there was no interaction between H4 and CDC7. Therefore, we can say S4 open isolation is probably important for H4 interaction with DBF4. Of course, we have some individual data, including PLA assay and chromatin and co-IP experiment from chromatin fractions. We don't have much time for that. So, and furthermore, to comprehensively investigate the function for H4 S47 open isolation in origin activation, with great help from Li Guohong's lab, we performed nascent strand sequencing. NS signal was increased by PUG, but decreased was due to the H4 S47 mutation. And once S47 was mutated, the PUG-induced upregulation was also blocked. Again, this gene-wide analysis further confirms the important role for H4 S47 in the regulation for origin activation. Given that open isolation is a nutrient sensor, which is able to sense the changes from nutritional supply, we continue to investigate whether H4 S47 open isolation can play a role in coupling gene replication to its nutritional environment. We can see so the replication deficiency in H4 wild type cells, when the glucose and the glutamine was reduced, replication efficiency in H4 wild type cells declined gradually, but remained rather stable in H4 mutant cells, indicating that H4 S47 open isolation might be important for the regulation of DNA replication in response to nutritional stress. In eukaryotic cells, how licensed origins are selectively activated has been a long-standing question. In this work, we demonstrate that H4 S47 open isolation is required for efficient DNA replication. Instead of origin licensing, it regulates origin activation. The establishment of open modification at H4 S47 by OGT could facilitate the loading of DDK complex to chromatin and in turn promote MCM, the phosphorylation of MCM complex and origin activation. Eventually, acknowledgement. So heroes in the labs in the lab are here. Yingyi and Jiayao are the main driving force for this project, and Ke Jian and Jingjing also made great contributions along the way. And I also want to take this great opportunity to thank Professor Wei Min, Li Guohong, and Long Haizhen for their support and contributions. Thank you all for your attention. Yeah, that's
okay.
Pretty much.
Okay, thanks. Okay, any questions?
Have you done the reverse pull down by using the DDK to see whether you only pull down the GlackMac form, but not the SA form?
That's a very good suggestion. We didn't do that, so that's something we should do. And actually, for this interaction, we have a lot of extra evidence, including the PLA assay and also purified proteins from other systems. But I totally agree. It's important to do it in the other way around. Yeah, thank you.
Hi, I have a question for your H4 study. As we know that the histone modification occurs mainly at the N terminal of H3 and H4, which is a tail.
Yeah.
Well, I think maybe S47 is in the globular domain. So also localization is a very big modification. So is there any change after this modification occurs in nucleosome level or the octamer level?
Wow, that's another great question. Yeah, this is very important to see whether it can affect the structure of the nucleosome or protein level. That's a very good question. So to be honest, I don't have results for that. So definitely, that's something we want to do, and we are trying to establish the system which can help us evaluate whether the role of H4S47 open oscillation in this kind of control. So it's very interesting, but we don't know. Sorry.
Any other questions? Any questions from online? Okay, so I have a question. I'm wondering. You take the organelle modification on H4 until H4S37. So does H4 until exist other histone modification, and if the other modification exists, can this modification have a competition role for the replication?
That's a very good question. Yeah, because as I told you, so this open knot modification usually takes place at serine and threonine, and we know serine and threonine can be phosphorylated. So, and also it is known that it is possible this site can be phosphorylated. So we also checked, and it is clear that in some cases the phosphorylation and open oscillation can compete for each other. And in this work, we also checked whether this H4S47 can be, whether the phosphorylation of H447 can play a role in gene replication. So we tried to synchronize cells and release them into S phase, as you can see here. So we can see during S phase that the phosphorylation of H447 wasn't there, indicating probably H4S47 phosphorylation is not that essential. We also tried to make other types of mutations, including 47D and 47E, to mimic the phosphorylation. And as you can see here, similar to H4S47A, mutating S47 to D and E also affect repressed DNA replication. So it doesn't matter whether it is phosphorylated or not. So the effect is the same, indicating phosphorylation might be not that essential for DNA replication. That's our idea, thoughts. Yeah.
So maybe I don't express my meaning clearly. I want to ask if exists other modification on other sites on H4 until.
You mean You mean crosstalk on H4?
Yeah,
so, yeah. As you mentioned, H4. Of course, I mean the H4 K20 methylation and also all the other types of modifications on H4. There might be some sort of some kind of cross talk between these kind of modifications, and it is typical. It is, I mean, for some other processes, cross